Okay, good morning, good afternoon. Welcome to Biomass Canada Cluster Project Updates webinar series. My name is Mahmoud Abadian. I'm a research associate at the University of British Columbia. I work on biomass and bioenergy logistics and supply chain. And I work with two research groups at UBC, including biomass and bioenergy research group, as well as biotechnology and forest product research group. As you may know, we have started this webinar series about 4 weeks ago. We already have updates from 4 projects. And today we are going to hear updates and progress on 2 other projects that are led by Dr. Kevin Vesey and also Dr. Lini Tian. And her colleague, Dr. Julia Liu. But before I introduce the speakers, I just want to quickly, again, remind everyone about the Biomass Canada cluster. It's a cluster established in 2018, led by Biofuel Canada Network and with the financial support from Agriculture and Agri-Food Canada's Agri-Science Program, and also with the contribution from industrial partners. As you know, the focus of this cluster, again, is to promoting the use of agricultural biomass and waste for bioenergy, biofuels, and bioproducts. And we have 10 activities in Biomass Canada cluster, basically 10 projects. And in total, this project, they look at biomass production, we look at the biomass pre-processing, and pre-treatment, and also we look into logistics and supply chain. So we make sure that we produce a feedstock which is suitable for, again, for low carbon bioenergy, biofuel, and other bio-based products. So as I mentioned that we are going to hear from 2 activities, activity number 6 and activity number 7. Activity number 6 is led by Dr. Kevin Vesey. He's a professor of plant biology at St. Mary's University in Halifax. And he's leading the project entitled Establishment and Yield Potential of 4 Biomass Crops on Marginal Lands in Nova Scotia. And then after Kevin, we are going to hear from Dr. Lini Tian, Dr. Julia Liu, and also Kevin is also involved in project number 7. Dr. Lini Tian, he's a research scientist at London Research and Development Center, Agriculture and Agri-Food Canada, and Dr. Julia Liu, she's a professor in the Department of Chemistry and Biology at Ryerson University. And they are leading activity number 7, which is about further investigation of sorghum for biomass on marginal lands, hybrid genotypes, growth promoting microbes, and plant usable nitrogen. So just quickly go through the housekeeping items. Again, if you have attended these webinars, you know that we posted recording and also a copy of presentation on Biomass Canada Cluster website on the webinar page. And also, if you have any question, please make sure that you put it in the Q&A box. And then after each presentation, we try to address as many as questions that we can. So with that, I ask Kevin to start his presentation. Thank you, Kevin.
Thank you, Mahmoud, and welcome everybody, no matter where you are in the country or in the world. Recognizing, okay, so make sure I get the right presentation up here. There we go. So I will be telling you about what's known as project number 6 in the Biomass Canada Agri-Science Cluster. And the title for today's talk is on the establishment and yield potential of 4 biomass crops on marginal lands in Nova Scotia. And this is done in collaboration with my colleague, Dr. Joseph Papadopoulos, from Agriculture and Agri-Food Canada. Joseph may be presenting a few slides. I'm not sure if he will be joining us in the presenting mode. Joseph, I sent you a link to connect to present just a few minutes ago. If you can find that in your email, you may be able to present your few slides, or I'll just cover that for you. Okay, so yes, and of course, I'm located at St. Mary's University in Halifax. So we start out with acknowledgements in this and a cast of thousands here. Of course, the Canadian Agricultural Partnership Program that funds the Biomass Canada. But in particular, I want to draw your attention to our many industrial and association partners here, all indicated here. And I just want to highlight the Nova Scotia Federation of Agriculture. A lot of this research is on-farm research with farmer collaborators within the NSFA. And I also want to highlight the Nova Scotia Innovation Hub, a fund here in Nova Scotia that helps is providing cold hard cash to this project, and their mandate is to stimulate the bioeconomy in Nova Scotia. So the goals of this research is to assess the potential of biomass crops as a major feedstock supply for bioprocessing in Nova Scotia. And so there's four goals in this. The first, to determine the yield potential of 4 biomass crops, the 4 shown here: 2 grasses, switchgrass and miscanthus, and 2 tree species grown as coppice, so coppiced willow and coppiced hybrid poplar. And these crops are grown on marginal agricultural lands. Specifically, because these crops can grow on agricultural lands, you don't have to use lands good enough to grow food crops. And so that's one advantage of these. We're also attempting to identify locally sourced biological inputs to enhance the growth of these crops. The margins on these crops are quite small, so you want to keep the input costs as low as possible. It really doesn't make a lot of sense to fertilize these crops for both economic and environmental reasons. But we've identified some locally sourced inputs that we're checking to see if they can stimulate the crops at low environmental and economic costs. We're also creating yield models. Now, we only have so many sites and there's not that much data on these crops in Nova Scotia. So we're trying to broaden this with a modeling exercise to predict the yield of these crops under Nova Scotia growing conditions. And finally, and particularly from my cooperation from Dr. Papadopoulos, testing the adaptivity of miscanthus, a variety of miscanthus and switchgrass cultivars to Nova Scotian conditions. So why are we doing this? Well, there's actually a lack of biomass in Nova Scotia for bioprocessing. There's many uncertainties about Nova Scotia biomass feedstock basket, as we call it. When people think of Nova Scotia, we're covered with a lot of trees. There's a lot of trees in Nova Scotia and a lot of rocks. But in fact, there is a limited volume of woody biomass in the province for many reasons that are too complex to go into here, but we're always, everybody I know who's processing biomass is always looking for more. Also, relative to a lot of jurisdictions, the wood that is produced, the wood residues, really not the timber, but the wood residues produced are at a high cost. And there's a risk of insecure supplies and seems to be changes in forestry policy. And so because of these first 4 bullet points, we're focusing on this last one, trying to address a gap in data for alternative biomass sources like these biomass crops grown on marginal agricultural lands. And from that point of view, Nova Scotia may have an advantage compared to some jurisdictions. Nova Scotia has an abundance of underutilized marginal agricultural lands that you could grow these crops on. This map shows our agricultural lands. Many of you may be familiar with our classification system 1 through 7 in Canada. For agricultural lands, we essentially have no class 1 land in Nova Scotia. That's the best land, should be, in my mind, reserved for producing food crops. We do have some very limited class 2, quite a bit of class 3, and a lot of class 4. And when you get into class 3 and class 4, these are lands that have starting with moderate to moving on to severe limitations in terms of production of food crops, but you can grow these biomass crops. So I say we're blessed, in quotation marks, with abundance of these sorts of lands, over 400,000 hectares of class land, for example, that we could grow. This is agriculture class land. You're not diverting land from other purposes, but basically right now it's generally underutilized. And some other advantages of these biomass crops, aside from they can be grown on these marginal lands, or maybe because they can be grown on these marginal lands, there's no of the so-called food versus fuel debate. So we're not diverting top quality lands that you could be producing food crops for fuel, so it avoids that debate. Also in Nova Scotia, and this might be more particular in Nova Scotia, but I'm sure exists in some other areas, we have quite a forest harvesting debate here in the province. A lot of the public have problems with forestry, almost for whatever the reason, whether it be for lumber, pulp. They really, it's very much of a conservation mode in Nova Scotia. So again, we're not using forestry products per se, we're using agriculturally classified land, just very underutilized land. Another advantage, all these crops are perennial crops. So unlike annual crops, many of the so-called first generation biomass feedstock crops like corn and wheat and sugar beet, perennial crops, once these grasses and trees are established, they can be harvested for many years. The grasses every year for up to 15 years before you should cycle into something else for crop rotation reasons. And the trees, you harvest them every 3 years for up to 30 years. Most importantly, you can produce high yields, 12 to 18 dry ton per hectare per year, with little or no inputs. So a lot of above ground biomass from on poor lands with very few inputs. And aside from all this above ground biomass that we're going to make, hopefully to offset the use of petroleum for biofuels and other bioproducts, below ground, what's happening is all that biomass can increase the carbon organic matter content of soil. Some of this organic matter going into long-term soil carbon pools like the humic fraction, hence sequestering carbon. So you're getting double duty here, both the biomass above ground going to biofuels and bioproducts, and the underground biomass helping to contribute to long-term carbon sequestration. And so if this is such a great idea, why isn't everybody doing it? And I refer to this as the chicken and the egg problem. So on one side, I don't know if it's the chicken or the egg, we have agricultural producers. I work with many farmers, and they can see that these crops can grow well on their marginal lands. But they say to me, that's great, Kevin, but I'm not going to grow these crops unless I have a market for the biomass, which makes perfect sense, because why would you invest your land, your time, your money in establishing these crops if you don't have somewhere to sell the biomass in the end? And then on the other side, again, whether it's the chicken and the egg, you have the biorefiners or investors that would use this biomass saying, well, we're not going to invest in a biorefinery to the tune of tens or hundreds of millions of dollars until they can be sure that they have a supply chain of biomass to meet their needs. So that supply chain, the biomass must be at the right quality, the right quantity, and the right price. So really, we got this inertia, this chicken and the egg problem. So our research hopefully will help reduce some of this investment risk for both producers and users of the biomass by determining the yield potential and some aspects of economics with this research. So what we're actually doing, as I said, we've got sites all across Nova Scotia, 7 in total. They all have the same basic design for you agronomy geeks out there. And what we're looking at here is a replicated block design. So 4 blocks, 1, 2, 3, and 4. And then within each block is each of the crops: switchgrass, poplar, willow, and miscanthus, randomized within the block. And then within each plot, there's subplots with the amendment treatments, biofertilizer treatments, we call it. So the BF stands indicates. So we have the pulp mill residue, an anaerobic digestate, and a seaweed extract, and then an untreated control. So we'll talk about that a little bit more. That's the basic layout. So this is replicated 7 times from Cape Breton to from the northern part of the province right down to near Yarmouth in the southern part of the province. And in fact, here's where they are. So there were 5 sites put in 2019. Those are indicated with the red marks here in the more the northern half of the province. And then we were supposed to put in more sites in 2020, but a little thing called a pandemic got in the way of that. So the last 2 sites, one here in Falmouth, Nova Scotia, and one here in Chicogan Point near Yarmouth, went in 2021. So we cover the province quite well, quite a diversity of both meteorological and soil conditions across these sites, but all on marginal lands. So what are we measuring? So we establish these crops, and then through the year, and particularly at the end of the year, we measure things like survival rates, plant height, plant area, stem and tiller number, of course, yield. We also look at soil and tissue nutrient concentrations. We're recording meteorological data both on site or by nearby Environment Canada stations. And this year, we're also starting to measure, we're going to start measuring quality aspects of the biomass from these different crops at these different sites. And this is, I just want to show you a few of the important people here in my crew. This is Mon Fay, my long-suffering research associate, who really heads up the field team. And also pictured here is Dr. Not Dr. yet, Emily Manton. She is a PhD student. This is Cameron Dazell, who was a master's student, is now a part-time research associate. And then we've got a cast of thousands of undergraduate students and others that help out on the field crew. And so these soil amendments I was talking about, here's a picture of the wood fiber residue. We get this from our colleagues at Port Hawkesbury Paper. And this is basically essentially a waste product from the pulping, mechanical pulping process. And it's basically pure carbon, pure wood fiber. And it's basically a waste stream. So we apply that. We also have what's called a liquid fraction of an anaerobic digestate. So an anaerobic digester, these very large bioreactors, often associated with dairy farms, where you put in dairy manure, crop residues, all kinds of organic matter, and basically make biogas. One of the byproducts of that is this liquid fraction, which is a very, you see it nice and black and organic looking, and it smells a little funky, but really is a low grade fertilizer, but also I'm sure has many biostimulants in there as well. And then finally here, a seaweed extract from a local company, Acadian Sea Plants, which is also known to be a biostimulant. So this picture comes from Miscanthus, one of our Miscanthus plots at Nepan. This is in 2019. This is just a few weeks after establishing the plants. And you can also already see some of the effects early on of these soil amendments. Here's the control plot. Here's what the plants look like that were treated with the pulp sludge, which is turned into the soil at planting, the digestate that's planted, supplied shortly after the plants emerge, and also the seaweed extract. So some evidence just visible there. When we actually get around to measuring this stuff, we can see effects, but I first want to draw your attention. So this is from one of our sites in Cape Breton in East Sky Glen. This picture was taken in September, early September of 2021. This is Miscanthus here and hybrid poplar on the right. So these crops are only two years after establishment. And look at the amount of biomass we're getting on the Miscanthus. This is cut back every year. So this is just the growth in this one season. And being a perennial, this will continue to grow into October, sometimes if it's a warm, even into November. And then the poplar, it's been growing since 2019. We only harvest it every 3 years. First harvest will be in 2022. So that's what the crops look like. A couple of more of our intrepid student assistants. And then on the right here shows some of the effects of these soil amendments on the dry weight of Miscanthus, actually from the site, East Sky Glen. And so we have the control treatment here and the dry weight in kilograms per hectare that arises from untreated Miscanthus at this site. And then either 1 or 2 applications of the liquid digestate. This is the pulp sludge treatment, and either 1 or 2 applications of the seaweed extract. And so again, you can see here, we're getting some, this is quite consistent across sites and actually across crops. The pulp sludge seems to be doing some very interesting things, increasing the yield here significantly. We see pretty consistently effects of digestate, not so much with the seaweed extract. It's more variable. Sometimes we don't see any effects of the seaweed extract. But there is a trend that we seem to be seeing so far. And this is just, again, more visual examples of this. This is the miscanthus at Nepan in September of 2021. And again, here's the control plot. You can see just visibly here the difference from the pulp sludge and the digestate compared to the control and the seaweed extract over here. So I always tell my students, if you can see an effect when you're looking at the crops, you can be pretty sure there's going to be a statistical effect when you actually do the analysis. And then this figure shows some results, again, looking at one of the crops, miscanthus. So we're looking at miscanthus yield here on the y-axis in dry kilograms per hectare. Across our 5 sites, so Sky Glen, Port Hood, Bible Hill, Nepan, and East Gore, the 5 sites established in 2019. And then each of the histograms represents each of those treatments. Again, the control, 1 or 2 treatments of digestate, the pulp sludge, and 1 or 2 treatments of the seaweed extract. But you can see we have the yield potential varies across site, Sky Glen being our best with almost 20 tons per hectare in a single year. Again, this is just 2 years after establishment. Yield potential really doesn't max out on these crops until 5 or 6 years after establishment. So quite amazing there. But contrast that with Port Hood, which is only about 30 kilometers away from the Sky Glen site, but it's much more, it's very close to the ocean, much cooler. This site has quite a heavy soil in it. I mean, we're still getting about 5 ton dry ton per hectare, but this is the max. We see closer to 20, the min around 5, and then the other 3 sites were intermediate. But again, generally this effect of pulp sludge and the digestate stimulating the growth of the crops at most sites. So that's what we're in the middle of in terms of the field studies. I also mentioned that we're doing yield modeling. There's just too few, even with our studies. There's just too few studies of the biomass potential of these crops to confidently inform industry of their potential in the province. So we took an approach of yield modeling, and how we did that is we looked at these biomass crops from studies around the world, but only in areas that had climates comparable to Nova Scotia. So basically in terms of the severity of the winter and their temperatures during the growing season. And we started out by looking at over 50 soil and meteorological factors that could be influencing these growth. And after a lot of work by my PhD student, Emily, she identified 2 factors, that is growing degree days, basically accumulated heat during the growing season, and precipitation during the growing season, that were strong indicators of to yield, to model the yield. And this was done originally in switchgrass. And so the output of some of that, one of the ways you can look at the veracity of your model is shown here. What we're looking at is the measured biomass yield along the x-axis and the predicted biomass yield based upon these 2 factors, growing degree days and seasonal precipitation. So what our model would predict. Each dot here is a site year of data from a study somewhere around the world. And so we're very pleased with this. You see a nice regression line there. The gray line is the 95% confidence interval, so very tight confidence interval there. The dashed line is what's called the prediction interval. Also note, I don't know how well you can see this on your screens, but the black dots are data points from studies of switchgrass yield from somewhere outside of Nova Scotia, and the blue dots are of yield of studies within Nova Scotia. And so we were very happy to see the blue dots well sprinkled among the black dots. They're not all grouped at one side or the other. That makes us, gives us some confidence that this will reflect, this model will reflect well of the yield potential of Nova Scotia based upon the few empirical measurements we have to date. And on the right here, we have an interesting exercise. We took our yield model, and then we took these two factors, growing degree days and precipitation, seasonal precipitation, at two sites in Nepean, Nova Scotia and Kentville, Nova Scotia. And we fed those into our model. And what would the yield be like of these switchgrass from 2011 to 2019? And you can see in any one year, we had the average was about 10 ton plus or minus 1 ton. The low here about 9.7, and the high up here above 10. But that's an interesting exercise to see, what the yield might have been. Unfortunately, we don't have enough data points to correlate this to actual measurements on the ground, but it gives you an idea of what the potential might have been which we have quite a bit of confidence in our model. So there we go. So another aspect of this study is the screening of both, and I see, I'm just checking here. No, Joseph is not in the presentation mode, so I'll just cover these slides for him. Joseph's background is both breeding and agronomy, predominantly breeding, and he has been screening developed cultivars or experimental lines of both switchgrass and miscanthus to try and see which lines might be best suited for Nova Scotia growing conditions. And in switchgrass is a productive and persistent bioenergy species in Canada, I'd say across North America, in fact. And there have been long-term evaluation trials of switchgrass in both the US and Canada, looking for high yield and winter hardy cultivars. Agriculture and Agri-Food Canada breeding program is making good progress on enhancing the persistence and yield of these switchgrass to be used as biofuel feedstocks and for other bioproducts. But so switchgrass study, there's quite a few to choose from there. Miscanthus less so, a limited number, and that will be reflected in the data that I'll show here. Okay, if this move forward, there we go. So this is actually looking at Joseph's data from switchgrass harvests. So yield here in kilograms, that must be tons per hectare. From 2019 to 2021, there's the long list of both cultivars and some possible experimental lines there that he's been testing. We're actually using this one in our test, the cave and rock. Is what's in our field study and across the province, but you can see some of the potential there up to 7 ton. Whoops, sorry, jumped there too quickly, but quite a bit of variability. So this work is really important to identify which of the germplasm out there in switchgrass might be best situated for Nova Scotia. And again, this sort of work may inform where these crops would grow well in other regions in Canada. And then, as we said, fewer lines of miscanthus to choose from here. A few lines being 3-4 lines being tested here. And again, you can see quite a bit of variability from some not performing very well to some doing quite spectacularly. Okay, so that's an additional work. I should say Dr. Papadopoulos, of course, is also collaborating on our field studies of the 4 biomass crops. One of our sites is at Nepean, and we couldn't do it without the cooperation and collaboration of Joseph and the staff at Nepean Research Station. So we're asked to talk about some of the relevance of our work, and this is very practical work. So some of these messages are very practical messages for what my farmer, producer, collaborators want to know. And some of the things we've learned about establishing the crops. And one is that if you got a lot of these critters around, deer, they become a serious issue for establishing tree species. So this is from our site at Bible Hill, and the neighbors there love chewing on the tree species, the new growth that comes out in the spring. And to the extent that we had to put up a deer fence around our site, or we probably wouldn't have any trees to be measuring, any of the yield to be measuring. Of course, that's not practical to scale up on a commercial level, But the take-home message here is if a producer has a kind of an out of control or abundant deer population on his or her land, he or she really probably should avoid the trees and stick with the grass crops. We've also discovered, I mean, we were told this in the on the outset, but it came all the real whenever you do it, is that weed control is extremely important in the first couple of years. In some crops more than others. The miscanthus filled out the canopy very quickly. We'll talk about why that is. Switchgrass less so, to the extent that although we didn't want to use herbicides, we really, in the second year, we did apply a domestic herbicide to knock back the weeds. Otherwise, we were afraid we were going to lose the switchgrass plots. So that's another thing. So this is something to be worked out. We did all our weeding and all the other crops mechanically, essentially by hand or small machines. Again, in terms of a scale up, an effective weed control measurement is absolutely necessary in the first 2 years, I'd say. And then this was one that we didn't expect at all. But at one of our sites, Port Hood, you might recall that's the one where we're having some of the poorest yields. And this is a quite heavy soil and also is quite cool site. And what we're quite surprised. So in 2019, these cuttings were planted. Go back in the spring of 2020. And what we often saw was this. So this is the original planting height of the cutting was here. So this was down here when we planted it. But over the winter, through frost heaving, this stem was basically pushed above pushed up through the soil, which often kills the cutting, or sometimes the cutting is actually pushed out. We'd find them lying on the surface of the soil, and of course they're dead at that point. So again, lesson learned: if you have a particularly heavy soil, the trees are probably not going to establish well. Better to go with the grasses. And then I've referred to a fantastic growth in my mind we've been having of the miscanthus, and I got to say personally, this is one of the biggest, most pleasant surprises we've had from the research, is how amazingly well the miscanthus is doing. And the context for that is that miscanthus had, the establishment and growth of miscanthus had been tried several times in the province before with mixed results, generally negative results. And we've been having fantastic establishment and yield early, the first few years of yield. So why is that? Well, the big difference, I believe, is that these earlier studies and how miscanthus is often started is from rhizomes. So basically underground stems associated with the roots that are harvested and then replanted, which is very labor intensive, and the rhizomes are not cheap to start with. But we had tried that and didn't have much success. Might get 30 to 50% survival for all that work and labor. And so my research associate, Dr. Fay, who I called the tissue culture wizard, said he thought he could make tissue culture from these apical meristems on the rhizomes from the plants. And sure enough, here there are the buds growing in culture media, growing out to plantlets, which we then transfer into the greenhouse, grow them up in the greenhouse, and then we transplant them. And within a few weeks, you've got a nice thick crop with essentially 100% establishment and 100% overwintering survival through that first winter. So this may be the big difference why we're having such success with miscanthus. Now, the big question is this economically feasible? Now, planting rhizomes, and rhizomes are generally about 50 cents a piece, so they're not cheap either. But we're actually doing an economic analysis to see whether this is economically feasible. We definitely know that it is technically feasible and agriculturally, agronomically is successful, but we'll see whether it is economically feasible. My tendency is that yes, it could be. Okay, so some of the success outcomes we've been asked to speak to. So in general, this research will determine the yield potential and identify best practices for the production of biomass crops in different agriculture areas in Nova Scotia, thereby reducing the financial risk of potential producers and users of the biomass. We hope it may help create new revenue streams for agricultural lands for producers for agriculture producers from these underutilized marginal lands. May help diversify biomass feedstock base to help attract more biomass processing facilities. And finally, particularly for the pulp mill residue and their anaerobic digestate, might identify new markets for the sale of these products. Critical success factors, basically a lot of it comes down to economics. Will the yield potential be high enough and the cost of production low enough to convince agricultural producers to grow these crops? And will it be attractive enough for biomass processing facilities to move to Nova Scotia, or more of them? And another success factor, as is seen in most jurisdictions around the world, we need government policy to help grow this aspect of the bioeconomy in this area if it is to be successful. So future work, we'll continue to maintain and collect the data on our 7 established sites. We will finalize the yield prediction models for all our crops, complete our economic analysis of the miscanthus from tissue culture. This year, we're starting on getting serious about testing biomass tissues for quality characteristics, and Dr. Papadopoulos will continue testing the switchgrass and miscanthus cultivars under Nova Scotia growing conditions. So in summary, I hope I've showed you that using on-farm trials, yield modeling, and testing cultivars, we developed a good understanding of the yield potential of these 4 biomass crops grown on marginal lands in Nova Scotia. We've shown that these crops can be established and have significant yield potential in their first few years of growth. And we hope that the outcomes of this research should reduce the investment risk to both biomass producers and biomass users. We've established that these crops can be established and produce significant biomass on marginal lands. The commercial success of the use of these crops will be dependent on the development of transport systems and markets for the biomass. I haven't talked about this at all, but other presentations in this webinar series does talk to the supply chains and logistics of getting the biomass to the processing sites. And that is an important aspect, but something we've not looked at. And in the next year of work, we will characterize better the value of these crops and do dig deeper into economic analysis, particularly of the miscanthus plantlet production. And with that, I'll end and I'll end with special thanks to all these many contributors to the research. I'll stop sharing my screen there.
Okay, thank you very much, Kevin. You know that the demand for biomass, Kevin, is just growing globally, even in Canada, in the US. And now we see that many people come and ask about the opportunity of dedicated biomass crops in Canada. So I'm actually very glad to see that, Kevin, you're leading the way, at least in Nova Scotia, on the opportunity of not only one, but a couple of actually dedicated biomass crops in Canada. So thanks a lot for that. We have about 5 minutes for a Q&A, so if you have any question, please put it in a Q&A box. But I kind of start the question, Kevin, is that, so you look at different biofertilizer, or you call it soil amendment. Do you think these soil amendment, they are going to impact the ash content when you harvest the miscanthus? Do you think, I'm not sure if you have any work on this space, but do you have any idea these type of biofertilizer, they might impact the ash content of miscanthus or other type of dedicated biomass crops?
Really interesting question. The answer is, I don't know. That we will know more after we do our analysis of our tissues from the next harvest this fall. But my gut says no, because if you think about ash content, and I'm not a chemist, but the ash content, it's really the mineral components, right? And when we look at that wood fiber residue, it is just carbohydrates. I mean, it's just cellulose, lignocellulose, lignin. That's basically what we're putting in there. Now, there would be some, what is the ash content of that? Would that contribute, and then how would that affect the overall uptake by the plant? Those are a lot of questions. And the digestate, like I say, it's basically a low quality fertilizer. We do have a chemical analysis of both of these, but there's one thing to see the chemical analysis of the input and then what actually ends up in the plant. So I'd hold off making any conclusive comments on that until we do our deeper analysis from the harvested material harvested this fall.
Okay, no, sounds good. Well, actually, the reason I'm asking is that, as you know much better than me, at least I've worked on miscanthus because that's why I'm mainly talking about miscanthus, is that the ash content is quite low compared to other type of biomass. So that's what I'm just curious that if we use different fertilizer, how they're gonna potentially, again, I have no idea either. So how they're gonna impact the ash content. So that's just a question I had in my mind. And I guess we have again, maybe the second question. Let's see if I have, yeah, I get Julia actually, she has a question. So she's saying that you mentioned that you measure soil and tissue nutrients. Do you measure nitrogen as well? And if yes, do you see any correlation between nitrogen in soil and in tissue?
Ooh, yes, we measure nitrogen. Our soils, because they're marginal, and they had been cropped generally with low quality forage, unimproved pasture. Those are the sorts of sites that the nitrogen levels in the soils in general are quite low. I can't tell you off the top of my head whether there is a strong correlation between soil levels and nitrogen and soil level nitrogen and tissue level nitrogen. We are seeing definitely stimulating effects of things like the pulp sludge, the pulp mill residue, and the digestate. But if you look at the amount of nitrogen, there's almost no nitrogen in the pulp sludge and the wood and the digestate. It's about, I can't remember, it's 1 or 2%, if that. That's not a stimulating effect. What I think these things are doing are and we still have gain the evidence of this. It's really more, it's a soil health. So these are some of these things are quite poorly structured soils. So it's adding structure to the soil, water holding capacity. You put any carbon in the soil, you're going to stimulate microbial activity. That's where I think a lot of the stimulation are coming from our soil amendments. So we do see effects on yield with different soil amendments. But that correlation between all the soils were quite low in nitrogen, and they were not received no chemical fertilizer. So I doubt if we'd see a big effect of soil in on these growths, but we'll get into that deeper. We haven't really got into the sort of meta-analysis across sites very significantly yet.
Thank you, Kevin. Another good question from Alison. So she's saying that when you discuss use of marginal land, what was the study plot used for prior to this application? And have you done any work to analyze below ground soil carbon?
Okay, yeah, very good questions. So yes, as I mentioned, we specifically, when we went to these sites, now Nepan is a bit different because it is a research facility, and one of our sites is at the Dalhousie Faculty of Agriculture Research Facility again. So those had various crops in them, but all of the farm sites, we specifically asked for poorer quality sites. So most of them were in basically low quality forage, yeah, this idea of marginal land, you wouldn't be growing corn on these or soybean. The soils are just not good enough. So I forget the second part of the question there, Mahmoud.
That was about, let me just go back. Have you done any work to analyze below ground soil carbon?
Oh, no, but that is on our to-do list starting. Okay, yeah, it was not part of our in this cycle. Many of you know that we're hopeful that the Biomass Canada will be renewed for a second cycle, and that is definitely a component of our proposal, if the second cycle is renewed, is starting to look at the carbon storage capacity under these crops. There's lots of literature to suggest that. The level of it that's going on in these trials just hasn't been ascertained yet.
Okay, great. And I think Julia, she has a follow-up question, and also Yusuf wants to add some comments. So let's ask, let me ask a question from Julia, then I let Yusuf to actually add a few comments. So Julia, she's asking, did you measure tissue nitrogen along the growing stage? If yes, did you see nitrogen in tissue varying with plant age?
No, we didn't do that level of detail. We're not really, for us, it's really about end of season yield. So, I mean, we did do, I think in year 2, we did some mid-season measurements, and there is tissue that could be analyzed, but this was not meant to be a deep dive into the nutritional aspects of the plant. So sorry, we don't have that. Yeah.
Okay. So Yusuf, I think you can talk. Just make sure that if we just briefly, maybe in 3 or 4 minutes, so we can move on to the next presentation. Thanks, Yusuf.
Yes, thank you, Kevin. That was a very good presentation. I just want to point out from the first graph, it's ironic, I was present from the beginning, but nobody can see me, so.
Yeah, sorry, I didn't know if you could connect, or so anyway, let me bring up that graph for you.
Well, that's okay, no problem. Anyway, the first graph about the switchgrass, because of the time limitation with this presentation, I'm only showing the result here from a newly established trial. It was these cultivars were established in 2018, and as you can see, the establishment of switchgrass takes more than 1 year to show establishment, and there is a good deviation between some cultivar and others. So there are cultivar picking up quite fast toward a potential of useful potential. About 20 years ago, when we started thinking about biomass crops, we had a focus group where we brought key producers interested in these topics. And we wanted to make sure that if we get to grow this, is there a potential market for it? And after discussion and calculation, the producer told us, I am not going to be growing this crop unless I get 10 tons per hectare. And the bottom line is that we're looking for cultivar that can grow an following establishment to produce that 10 tons. Not only, usually we're beginning to see in third production year, we're beginning to see the yield around 10 tons per hectare. And we have a study that we didn't show the slide, that established 2018. We peak around between 12 tons per hectare with different cultivar, and the peak persistent is a key. So producers are going to invest in establishment, and they need to harvest it for more than, like Kevin was saying, 15 years or so. So I'm not sure if we have the right cultivar yet that can, after establishment, to stay with high productivity for 15 years. But this is what we're studying. On the last comment I want to make is on miscanthus. Kevin is right. Our biggest problem getting good establishment with the current tubers that we can purchase. In the slide you saw, we were able only to access 4 tubers, type variety tubers for miscanthus. And 2 of them did not persist in our region, didn't even grow. The other 2 established. And guess what, with COVID, we tried to get the one that grow well to put in a new trial, and they were not available. So the miscanthus availability for actual farm productivity is not still there. And Kevin and I have been talking quite a bit about his variety, because he uses plantlets transplanting in the field. And if the economics is feasible, he's right. When we put them in the ground, we see really good establishment. And lastly, we have a current study now looking at using the most effective way with limited resources, like particularly nitrogen, to input into the crop, so reduce production costs to produce a productive biomass crop, particularly in switchgrass and miscanthus. That's all. Thank you.
Thanks a lot, Yusuf. If there is any follow-up question, maybe after the second presentation, let's see how much time we have. So I guess we have to move to the second presentation by Linging and also Julia. And I guess Kevin also, you may have a few presentations on activity 7. Linging, over to you.
Okay, second. Yeah. Okay. Yeah. Good afternoon, everyone from Ontario. Here we presented the biomass cluster activity 7. The research to be presented today is for past years research progress, and activity 7 has 3 labs doing different part of the research, and myself and Kevin Vesey and Julia Lu, and each of us will present each lab's research activity, and I will start first. As you see, Kevin's presentation, and also from other source of information, Canada has a large area of marginal land, and this marginal land are usually not suitable for many other crops like wheat and corn, soybean. And sorghum is a plant which, compared to many other crops, they can adapt to different types of soil and also growth condition. So its adaptability is good. Then what we thought is we could grow sorghum on marginal land and produce biomass. As Kevin mentioned, in this case, there's no land competition with other food crop. And my part of research for the past years focus on sorghum or hybrid. Yeah, just quickly acknowledge, and yeah, this project is under AFC CAP program, and also we would like to thank the Biomass Biofuel Canada, who is leading this program. And this program represents an important aspect of agriculture, climate change, air pollution reduction, and also bioeconomy. And our industry partner is ARC company in Ottawa. my research lab team is Mimi Lu, Josie Kelly, and Rowan, and Sufan Tahai, and also Albert, and the farm manager of AFC, and his crew provide a lot of help. And the goal of the research is straightforward. We just grow different types of sorghum, different marginal land, and we hope to identify some hybrids which can produce high biomass marginal land. Yeah, the approach is straightforward and just grow plant. And then last year, we evaluated 3 sorghum hybrids, 10A118, 131, and CSH45. These 3 hybrids were selected from 2019 research. In that year, we evaluated a total 5 sorghum hybrids. These 3 hybrids performed better than the other 2 hybrids, and we would like to further evaluate these 3 hybrids on marginal land, because for this field study, we need multiple year data to confirm the results. And the site we used is Simcoe in Ontario and Ottawa, and also London. London is not a marginal land, it's a primary land and typical agricultural land, which is used as a control. One of the major factors we evaluated is nitrogen fertilizer. So nitrogen fertilizer was applied at 120 kilogram per hectare. Yeah, just a quick plot design. And for each hybrid, we had 6 mini plot. And for this 6 mini plot, 3 plot were applied with nitrogen, 3 without nitrogen. And total we have 18 mini plot. And this plot will arranged in a random way. Yeah. This is yeah, just plot preparation. Yeah, showing some kind of photos. Yeah, so plant development. And one thing I want to mention is on the right figure, you can see between the sorghum plot, on the border of each plot, we grow the millet. The purpose is, of growing millet is to prevent the weeds growth. Because in 2019, we found the weeds growing is wildly, it's very hard to control. And we grow this millet and to control the weeds control. And the millet, we wouldn't allow the millet to grow too high. And we once it grows certain size, we cut them down and to maintain the plot, you see, and sunshine and airflow. And one of the major factors we study is nitrogen fertilizer. Because the big title of our project is to develop high biomass production on the low cost. The nitrogen is usually is the #1 cost for many crop production, and it's also important input for sorghum. So we would like to see how we may able to optimize the nitrogen fertilizer and to maintain a high biomass crop for sorghum. And what we did is we applied the nitrogen fertilizer twice. One is when the plant was about 2 to 4 inch high. The second application is when the plant was about 2 feet high. The total rate for the growth season is 120 kilogram per hectare, which is a kind of typical amount of nitrogen fertilizer used for like sorghum, like corn, and we just use that kind of representative amount of nitrogen to give to test. Yeah, so just show a picture and this the plant developed and very nicely. And when about half of the plant started to flower, then we measure the plant growth. We measure 3 part. One is plant height. For plant height, it's from above ground to the flower part. Exclude flower, and also measure the on-site measure plant fresh weight, and also we transport the plant back to the lab, and then we measure the dry weight, and after put into oven. Yeah, this is the showing some activity for plant harvest and measurement in our Ottawa field. So we, as mentioned, we measure the plant height, plant fresh weight, plant dry weight, but I only present the dry weight, which is probably better represented the real biomass production. And this is the dry weight in Simcoe field. So we can see the different hybrid perform differently. In this case, the right one, CSH45, showed better growth on Simcoe land. And regarding the nitrogen fertilizer effect, we can see the for 118 and 131, and apply nitrogen fertilizer has effect biomass production, but on CSH45, there's no nitrogen effect. This for the dry biomass in Ottawa. This one is different. You can see the still CSH45 showed the highest biomass production, but the nitrogen, applied nitrogen has no effect on biomass or dry weight production for all the hybrid. And this is the Lundell field, which is used as a control. In this case, you can see the 10A118 show the higher biomass dry weight yield compared to other 2 hybrid. Yeah, just a quick summary. And from the results, we can see certain sorghum hybrid grow better and produce higher biomass than other hybrid on marginal land, which means and in the future, if we do want to grow sorghum for biomass and select a specific hybrid is important. And then also the hybrid which grow well on typical agricultural land may not necessarily grow well on marginal land. Then in terms of nitrogen fertilizer effect, then what we thought is we know nitrogen is important, but why sometimes we don't see the nitrogen effect? Then we thought is based on the literature and based on the industry, then they said the sorghum has a very nice, very deep root system, and which they may kind of absorb just nitrogen in deep soil. So which means sorghum may kind of use nitrogen more efficiently, and so even if we don't apply nitrogen fertilizer, but from the nitrogen fertilizer residue in previous years, yeah, could be used by sorghum. If this is the case, this means grow sorghum kind of result in less nitrogen loss to the environment. In this case, nitrogen fertilizer usage can be optimized in the future to minimize the crop production cost. So this year, so what we plan to do is to evaluate more and new sorghum hybrid for biomass production, and we wanted to identify more sorghum hybrid that can produce higher biomass marginal land, and the information can be used in future program or by industry. Yeah. So I stop here, and the other two labs will share their results.
Okay, I'll jump in with mine then. I hope that you can see my presentation there. Just yell out Mahmoud if it doesn't. So yeah, so my work on this project, again with my colleague Dr. Yusuf Papadopoulos, is looking at the potential plant growth promoting microbes on these hybrids. So our research goal is to determine if some PGPR, plant growth promoting rhizobacteria, and actually we tried one rhizofungus as well, can stimulate the growth of hybrid sorghum genotypes on marginal lands. And just as a way of an overview, work done in Dr. Linning's lab before has shown that one very famous plant growth promoting rhizobacterium, Gluconacidobacter dasytrophicus, can infect some sorghum genotypes. This bacterium is known to stimulate the growth of a wide range. It was originally discovered in sugarcane, but has been shown to stimulate many different kinds of crops. Work out of my lab as well showed that various PGPRs can stimulate the growth of various biomass crops. So we've done work with hybrid poplar and switchgrass, with sugar beet, and with miscanthus, and in each case found various bacteria and sometimes fungi that can stimulate the growth of these crops. So our approach was to use greenhouse and field studies with combinations of sorghum genotypes, both cultivars and some breeding lines, those test breeding lines that Linning referred to, and these plant growth promoting rhizoorganisms for their potential enhancement of plant growth at 2 levels of N fertility. We use the same levels of nitrogen fertility used in Dr. Linning's Tien's work. In one greenhouse study, and then this year in the field, we also will be using the 15N isotope dilution technique to determine if there's any nitrogen fixation. Several of these microbes have nitrogen fixing potential, and this test tells us if they're actually doing something with these plants. So as I said, we do greenhouse and field studies in 2019 and 2020. We did greenhouse studies, and in total we looked at 4 different genotypes, 2 cultivars and 2 test lines, and 5 different bacteria, the Diazotrophicus that I referred to, also Gluconacidobacter azotocaptans, 2 Azospirillum lines, Lipofrenon N7 and Brasilense N8, and this is one fungal line, Penicillium blei. From that work, we then when we went to the field, we narrowed it down to 3 test genotypes, again, CSSH45 and the 2 experimental lines, and 4 microbes were tested, Again, the Gluconoacidobacter diazotrophicus, Sodocaptans, and the 2 azospirillum lines. So the field experiment, again, using microplots. These are just 2 meter by 2 meter. So in a completely randomized design, 3 genotypes, 3 microbial treatments, and 2 fertilizers. In 2021, we had 4 replicates for a total of 72 plots. So that's the overall layout. We use the same nitrogen fertility and the same density, planting densities used by Dr. Tien. Going back to some of the early work in the greenhouse, I just wanted to highlight here. Here are the microbes that we tested. The 2 Gluconoacidobacter lines, DS1 and PAL5, 2 azospirillum lines, and the fungal line. And to make a long story short, it was only the PAL5T here, the gluconoacido line, that was significantly different than the uninoculated control plants. We were not surprised to see this of these various lines. There's probably the most in the literature of Diazotrophicus being able to stimulate the growth of various crops. So that was hopeful. However, when we took it to the field in 2020, and I show this title says nitrogen fertilizer. Actually, this graph only shows the treatments without N fertilizer. We did see effects of nitrogen fertilizer stimulating the growth of the crops overall, but for the effects of the microbial inoculants, there were no significant effects. Now, that being said, I hope I can show a potential trend here, and that is, if you look, here are the 3 genotypes, so the cultivar and the 2 test lines here below. And if you look here, in each case, we've got control, the DS1, N7, and N8. And there's this, what seems to be a pattern of increasing yield, just measured as grams per plant, individual plant, increasing as we go from the control through these various PGPRs. So although no significant effects overall, this trend gave us some hope that we might be looking at something here. Then in 2021, we again did these tests in the field, again with the 3 genotypes of sorghum, we decreased we only used 2 inoculants. That's the azospirillum N8 and the Gluconoacidobacter PAL5. This is in minus nitrogen and plus nitrogen. Again, you could just see here visually, we did get compare these, did get effect of fertilizer, not surprisingly. But again, we held out some hope that but there was no effects of inoculant overall. There was a lot of variability here. Some of this, the quality of the seed that we got, again, it was an impact of COVID on the seed production, was not the best. But I just want to highlight here, again, a potential trend. Here is the uninoculated control. Here's the azospirillum line, and here's the Gluconoacidobacter inoculated plants. And again, we actually just haven't had time to go in and do a specific analysis of within this one cultivar at this 1 nitrogen level to see if there's a significant effect there. So nothing, hints at some possible effects, but nothing statistically significant identified yet. So the relevance of this, hopefully, if something does work, we'll determine the potential of stimulating the biomass of these crops on marginal lands. If these PGPR are found to effective, we may be able to stimulate the growth of the plants with little or no nitrogen fertilizer. Critical to the success will be, although PAL5 has had some indication that original greenhouse study and some trends in the field, we need clear and consistent effects of this PGR on enhancing sorghum yield before you can go on. The big step would be develop a commercial inoculant for this crop. But we need, in my mind, we need stronger evidence before we start going there. And in future work, basically, we're going to repeat the experiment that we did last year. again, gluconoacetobacter pal-5 and azospirillum N8 with the 3 genotypes. So basically the except we will be using N15 to see if there's any, if there is a stimulation of pal-5 or azospirillum brasilense is also a nitrogen fixer, is any nitrogen fixation going on. And I think that is it. Yeah, just the summary here. Well, so brief, don't need to go over the summary of what I just told you. And again, special thanks to all these people that helped make this research happen. Okay, that's it for me. I'll stop sharing. And I think it's over to Julia.
Thanks, Kevin. Sharing to. Can you see it?
Yes.
Good, thanks. Hello, everybody. I'm Julia. From now, we'll officially change the name from Ryerson University to Toronto Metropolitan University. I would like to start with thanks the researchers who have been working on the project. They are all graduate students here, Wei, Tim, and David. Tim and David graduated. I also like to thanks Dr. Van See's group at the Samaritan University for collecting soil samples for us from the Leping sites because we couldn't go. Now our goal is to, to study the effect of the bacteria. It's mainly gluconacetabacter. It's long. And the plants and the fertilizer application, water-soluble nitrogen species in soil during the growing season. The reason for us to do so, because nitrogen, as all we know, it's important for plant growth. Here in this project, specifically, it's a sorghum here. Now, sorghum get nitrogen from soil. And plants usable nitrogen species are water soluble. So that's why we are doing the measurement of water soluble nitrogen species during the plant growing season. For doing so, we collect soil sample from 4 sites as marked here, 3 in Ontario, 2 are here close enough to show where it's from Nova Scotia. We collect the sample of plots as a background nitrogen. And on the plots, before or after, on the plots, after fertilizer application, we name it as after planting, and also on the plots at harvest. And experimental design is by the PI and Dr. Visay's group over there, and basically different hybrids, fertilizer application condition with and without, and some microorganism. And this slide shows the map of the sites, the plots. Here is for London and Simcoe, and here is for Ottawa, here is for the Leping. And those star marked background, on each plot, we collected 4 cores of 30 centimeter, as shown as the star. Then each core is divided into 2 sections from 0 to 15 and from 15 to 30 centimeters. And the sample from the field was shipped to core and stored in freezer until analysis. To start with analysis, the sample was thawed, then physically mixed, then passed through a 2 millimeter sieve. Like this, that's a prepared sample. Then part of the sample is used for moisture and total organic matter measurement. Another part is used to extract the nitrogen species using water of pH 5 and end over end mixing for 18 hours. It's mixture of two phases. Then the phases get separated using centrifugation, and the clear solution is used for analysis of nitrogen species. Using colorimetric method for nitrate and nitride, it is different color nitride because it's not directly usable by plants. We include it here because it is an intermediate of the nitrification process. So for analysis of these two species, start with nitride. This species of nitrogen is reacted with a reagent to form a colored species. And the concentration inside is proportional to the color. Then it's measured by UV vis at this wavelengths. That's for nitride. For nitrate, only need a pre-treatment to convert nitrate to nitride, the same analysis. Now the calibration curve is showing here, QA/QC control, including standard control chart and blank control chart along with the process. For ammonium analysis, also use colorimetry method, but the only difference here is ammonium is reacted with different reagent to form a compounds of different color. So it's measured at the different wavelengths. For calibration, as shown here, now I try to change the background color, but this color shown, but this is not that visible. You have to look at carefully. And the control chart is similar or the same way for nitrate and nitride. Now, the limit of detection for the species are listed here. It's defined in this way. From the number of samples analyzed, show that majority of the sample have the concentration of nitrate above limit of detection, and the majority of the sample have nitride and ammonium lower than that limit of detection. Therefore, the upcoming results will be focused on nitrate only. This slide shows nitrogen concentration in soil that's through the growing season or during the growing season from off plots. At scale, I want to bring attention to is at maximum 20. And here shows after planting, we use a scale of 50. It's better to use the same for comparison purpose. And after planting, the concentration of nitrate in soil has increased mostly, except here and here. And at harvest, most places say decrease in the value of nitrate in soil, except here it's synthesized, here is the pain side under one of the treatment. Statistical analysis is shown. Let's focus on marginal land, the bottom three row here. Soil depths, so nitrate concentration in soil is significantly different from the two natures we collected. And hybrids do show the difference at all the marginal land side. And for fertilizer application, two sites show significant difference, but not at the Simcoe site. The same goes to the temporal effect. So Simcoe has showed no significant effect. Compare marginal land and control side, that's agricultural land, there is some difference. Bacteria treatment only in the pain side shows some difference. Here we compare the nitrate concentration in soil sample between 2019 and 2021. These 2 years, the rate of fertilizer application is quite different. So in 2019, 45, and in 2021, it's 120 kilogram per hectare. And up panel here shows the value, background value. And this time, I believe it's the same scale. This is after planting. As you can see at the beginning, there's no difference between the 2 years in terms of background, even at each site, the actual area used is not the same, but it's side by side. And after planting, that's after fertilizer application, higher rate of nitrogen application did not really consistently increasing the nitrate concentration in soil. Here's a higher, but many times, many places is lower here. And similarly, it's observed that at both year at harvest, the nitrate concentration decreased to the background level at harvest. Now, statistical results show after fertilizer application, there is no difference between these 2 years result at all sites. And after planting, that's after fertilizer application, we've seen the difference at London site and at Ottawa site. There is no Pain site because that year there is no field study. So that's only these 3 sites are compared here. As you can see at harvest, there's no difference in nitrate concentration in soil between those 2 years. By summary from the result obtained for 2021, higher nitrate concentration in soil, in top layer soil. Then nitrate concentration increase after fertilizer application, but decreased to, in most cases, to background level at harvest. And in terms of bacteria treatment, the concentration of nitrate with this treatment showing the concentration in soil is higher than the treatment with this bacteria with this hybrid. That's after fertilizer application. Now, compare these 2 years of data, show the rate of nitrogen fertilizer application has no significant effect on nitrate concentration in soil. So this year will be focused on, we will be repeating the experiment to confirm the research result or the observation we get from 2021 collection. And as Dr. Tian mentioned, there are some new hybrid will be introduced. There are some new hybrid, sorghum hybrid. We'll do the test of the soil nitrogen for those new hybrids. Also, we'll be doing or enhance information exchange activities for the past years. So we have tried, but in most cases, due to COVID-19 pandemic effect, most presentation is done virtually. I'm hoping in the coming year, we'll be able to do presentations in person to enhance the effectiveness of information delivery. I think that's all my presentation. Thank you.
Okay, thank you very much, Julia. I guess we passed the session, but I guess we can spend maybe 4 or 5 minutes to address 1 or 2 questions. Julia, we have 1 question from Yusuf, or maybe that's for Lining. Yusuf is asking, what is the soil classification where the sorghum trials conducted in London and Ottawa?
Yeah, and as mentioned in the presentation, the soil in London is typical, is class 1 soil. Yeah, it's primary agricultural land, which is used as control, not marginal land. In Ottawa, a site is a small area. We don't see the official number, but we used a land which is poor for other crop, and we purposely use land in that area which is not good for other crops. Yeah.
Okay, and Lining, you raised your hand. Do you have any comment you want to add?
Not for me.
Okay, yeah, okay. So I guess with that, we can end the session. Again, I want to thank Kevin, Lining, Julia, and also Yusuf for providing updates on activity number 6 and 7. Thanks a lot for that. And we look forward to the next session. That's going to be in 2 weeks. We're going to have update on 2 other activities. And again, we're going to post the recording and the slide after this webinar on Biomass Canada cluster website. Again, thanks to all the participants for attending and also for asking questions and engaging in the discussion. Thanks a lot, and hopefully everyone has a good day. Thank you.
Bye-bye.
Bye.
Bye-bye.